temporal retinal fields, and on the left side, you have, again, a nasal and temporal retinal fields, okay? Now, from here, these particular axons from these retinal fields will project out, leaving the eyeball, and they're gonna project inside these two structures called the optic nerve. So you have a right and a left optic nerve. This is your cranial nerve number 2, okay? And then the optic nerve crosses over in this particular portion called the optic chiasm, and essentially continues in both sides, both right and left side, and we call these structures the optic tracts, because now it belongs to the central nervous system, okay? Then those particular axons will project all the way to the thalamus, because remember, vision is a sensory information, right? And it's not smell, so therefore it has to go through the thalamus first before it gets directed to its primary visual cortex. So the specific area in the thalamus that detect visual images or visual information is called the lateral geniculate nucleus of the thalamus. You guys remember the medial geniculate nucleus of the thalamus receives auditory information, right, before it gets diverted into the auditory cortex. So from the lateral geniculate nucleus of the thalamus, it also receives visual information coming from the retina, and from there, it will project all of this information to the visual cortex in your occipital lobe, okay? So let's draw these particular pathways, draw these axons, these neurons and stuff like that, and see how they're projected. All right, so any visual images from the medial visual fields, both right and left side, as it enters the eyeball, it will stimulate the temporal retinal fields on both sides. Okay? So let's say over here on the right side, these images here, as it enters the eyeball, it will stimulate the temporal retinal fields on the same side. Okay? Whereas visual images coming from the lateral visual fields on both sides, as it enters the eyeball, it's going to project to the nasal retinal fields on both sides. Over here. Make sense? Okay. Now, these retinal fields or these retinal areas contain, you guys remember, photoreceptors, so rods and cones, and then the bipolar neurons and the ganglion cells, right? The ganglion cells all converge together, all the axons form a bundle of axons as they exit the eyeball to basically form your optic nerve. So the axons coming from the temporal retinal fields, as it exits the eyeball, is going to run along the optic nerve on the same side still. As it comes across this optic chiasm, it stays on the same side, and it continues on the same side of the optic tract until it gets to your lateral geniculate nucleus of the thalamus, and then it splits into two portions, a superior and an inferior portion. Okay? On this side, same thing. So the axons here exit out the eyeball, follows the same side of the optic nerve, follows the same side of the optic chiasm, follows the same side of the optic tract until it gets to the lateral geniculate nucleus of the thalamus. Okay. Electrical stimulation carrying visual images from the nasal retinal field, which detect the lateral visual, particular objects. So let's say here, as it exits the eyeball, it follows the same side, so it travels on the same optic nerve. But once it gets to the optic chiasm, it crosses over to the other side and continues to travel down the opposite optic tract until it gets to the lateral geniculate nucleus of the thalamus. Okay? Same thing on the other side. These nasal retinal fields, the axons exit out, travels along the same optic nerve, crosses over the optic chiasm, travels down the opposite optic tract until it reaches the opposite lateral geniculate nucleus of the thalamus. Okay? So far so good, right? Now another thing I need to mention to you guys, that in terms of visual images that you are receiving, whatever image you see in the environment, as it imprints itself onto the retina, it's flipped right to left and upside down. And then those images, as it's being relayed progressively to the visual cortex, once to the visual cortex, that image is flipped right to side up again, okay? And it's a weird thing where it's like it's flipped in the retina and it's flipped back again in the occipital lobe. Now, what you see in the occipital lobe are just pixels. And then we have millions of pixels in here, which basically presents a much, much higher definition than any TV screen that we can ever imagine. Okay, so that's the reason why we see everything all kind of congealed together. If something happens in the visual cortex, you will start to see pixelated images, which is very bad. All right, now, from the lateral genicular nucleus of the thalamus, this is where the complications kind of starts a little bit. I don't know if I have enough of these pins, so. So let's say from here, these particular fibers that project to this part of the lateral genicular nucleus of the thalamus will synapse with these neurons. And these neurons then project all the way to the visual cortex, okay? Over here, same thing. And these neurons will project all the way to the visual cortex. Whereas these particular axons here, when they synapse in the lateral genicular nucleus of the thalamus, they will synapse with these neurons. And these guys will also project to the visual cortex. Over here as well. Okay. Collectively, all of these axons here, they form a tract, and we call this the optic radiations. Okay? So we have optic radiation on the right side, optic radiations on the left side. Now with the optic radiation though, they are divided into two separate tracts. One is called a superior tract, and the other one is called the inferior tract. Okay, the superior tract. Let's just pick one of these colors. We may call it the superior tract. Let's say the green guys over here. We'll call it the superior tract. So superior tract of the optic radiation versus the inferior tract, or inferior tract of the optic radiation, okay? So now with vision, it's weird because it's already divided into medial and lateral, but once it gets down from this particular thalamus all the way to the visual cortex, it's further divided into superior and inferior portion of that image, okay? Because remember, the image is flipped right to left and flipped upside down, so therefore it has to have those. So the inferior tract will relay information from the superior visual area of the visual field, whereas the superior tracts will relay information from the inferior portion of the visual field. Okay? All right. Now that being said, once you guys do this, because you're gonna have to do this, I'm gonna give you a blank sheet of paper. You have to draw all this out, label it correctly, draw all the tracks adequately. Otherwise, you're not gonna be able to answer the questions I asked you. Okay? And the questions deals with what we call visual defects. So what happens when a patient comes to you as a clinician and says, oh, I can't see, when I look this way or that way or, whatever way. You have to figure out, because the patient doesn't know this. You guys know this. So based on what the patient tells you, you can predict where the lesion is occurring, where the damage is occurring along this overall pathway we call the geniculostriatal pathway. Okay. So what we're gonna predict is where these particular lesions are based on what we call visual defects. Okay. So with the visual defects, that's a lot. That's like 12 different visual defects. We're not gonna go into that depth. Okay. So I'm gonna give you guys 3 basic ones and a couple of extra ones for you guys to kind of think about. Now, before we figure out the visual defects, what we always start with is we will draw out 2 visual fields, okay? So the 2 visual fields are represented by 2 circles and divide it into medial and lateral side. Okay? Imagine this is the left side of your visual field, this is the right side of your visual field. Okay? That represents the medial, the right medial visual field, the right lateral visual field, the left medial visual field, the left lateral visual field. Make sense? What the patient sees from out here, okay, from their environment around you. All right, so now let's say if a patient comes into your clinic and the patient says, well, I can't see anything from my left eye, okay? So let's say let's say the patient says, yeah, you know what, my vision is so distorted, I don't have any depth. When I look out, I mean, I can see fine on this side, but as it gets closer to the left, it gets really blurry, and then if I look all the way to the left, I may not be able to see much. If I close my left eye, I can see fine. If I close my right eye, I'm completely blind, okay? Which means, on the left side, both medial and lateral sides are affected. So therefore, based on what the patient tells you, you now have to predict which pathway is basically, like interfered with, right? What's going on within this overall pathways where this neurological information is now being truncated or being sliced or being cut or whatever, or there's something interfered with that. So if it's on the left side, and they're saying the left medial visual field is affected, then all you have to do is follow the pathway. So the left medial visual field projects to the left temporal retinal field, which then is transmitted by this fiber all the way to the same side of the lateral geniculate nucleus. So therefore, somewhere along here, that fiber is cut, okay? And then over here, the patient also tells you the left lateral visual field is lost, which means that this left nasal retinal field is transmitting the information all the way to the opposite lateral geniculate nucleus. It's also being disrupted. So therefore, something happened right there. Does that make sense? Okay, so this is cut number 1. And when the patient has this, we call this monoocular blindness. And more precisely, we call this left monoocular blindness. So one-sided blindness, left side, okay? So this is left versus right, monoocular blindness. And you guys don't have to worry about these terms, because if I do ask you about this stuff, I will give you these terms, and you can predict what's going on, I think. I gotta check. I may not have it. But anyway, okay, so that's cut number 1. All right, so for cut number 2, a visual defect number 2, let's just say. So again, left and right side, okay? Patient comes in and tells you that I've been getting, chronic headache for the last 8 to 12 months. Okay, the headache is kind of, lingering all the time. It's not so bad that I can't function, so, it's not like migraine headache. It's just a constant headache and pressure. And it started maybe about a year ago, but then my vision has been kind of blurry around the periphery. Okay, I can see fine. It's like I'm looking into a tunnel, right? And then just around my visual area is just so annoying because I can't really see my periphery. Okay, so basically what that patient is telling you is that they have visual defects in this area. Okay, so both of their lateral visual fields, both on the left and on the right, are affected. And since these two areas are affected, the visual information from the lateral visual field projects to the nasal retinal field, which basically means these fibers that is crossing over at the optic chiasm is going to be affected. So the cut is this way, okay? What happens when you make the cut this way? You have total blindness. And we don't talk about total blindness, okay? If I ask you about total blindness, that's too easy. Right? So if ask you, okay, well, what happens when you have this particular cut right here? That's what you have to present to me. Now, this is called tunnel vision. The tunnel vision has another name. We call this bitemporal, because it's both temporal side, right? Temporal, I mean, it's weird, like they mix it up that way. So bitemporal hemi means half. Anopsia means visual fields. So half of the visual field, both on the temporal side, are affected. And therefore, we also call this tunnel vision. Okay? So, usually, what is the most common cause of this issue? What do you guys think is the most common cause of this issue here? Where the patient just comes in with some kind of bitemporal hemianopsia, assuming they have no injuries. Right? It's not like they got hit in the head or whatever the case is. Right? What do you guys think?
Stress.
No, not stress. Stress, you probably have just blurry vision overall. So most common, and because you have to kind of associate all the symptoms the patient has too. The patient also reports headache. That is constantly, it's chronic headache that's going on for at least 8 to 12 months already, right? There could be other changes also, but then that's an obvious visual changes. Now, if you guys think about it, here's the optic chiasm. On top of that is the rest of that brain mass. But below so on top of this is also your hypothalamus. Below your hypothalamus is your pituitary gland. So your pituitary gland, for some reason, is growing out of control. Let's say you have a pituitary tumor. That tumor projects downward and presses down on this middle portion of the optic chiasm. And because of that, the patient will present with this. Sometimes, right? Now, another thing that can happen is if this occurred in a relatively young female individual. Young female individual, maybe mid to late 20s, okay? Let's say clinical scenario, this young female individual that came to see you has no underlying medical condition. That's a clue to tell you there's nothing else that's going on with this particular person. So you can't say, oh, maybe she has this already happening to her, right? So healthy otherwise comes in to get evaluated by you as a clinician because she got into a car accident. Now the reason why she got into a car accident is because she didn't see the car coming from her side, and she's went through the light or whatever she did, or she turned and she hit that car. She got into a collision. Now you evaluate this patient, and the female patient is relatively young with this bitemporal hemianopsia. That is an early sign of multiple sclerosis for young female patients, and only young female patients. No young male patients, okay? If young male patients have that, maybe trauma, maybe some kind of pituitary adenoma. But for young female patients specifically, it is an early sign. It's not always a diagnostic sign, but it's an early sign that this could be multiple sclerosis, okay? Because relatively young people should not have any of these kind of problems unless you have some kind of trauma that's going on, okay? All right, anyway. But it's just something to kind of think about, because board exams like to kind of give you guys these scenarios. If you guys ever take any kind of standardized exams for anything, right, they'll throw these scenarios out at you. Okay, so let's say there's a third cut. So that's bitemporal hemianopsia. For a third visual defect that you guys need to know, it's just a basic one. Let's say if I cut here. What do you think the visual defects would be? So all we have to do, and that's the reason why you guys need to draw these correctly, okay? So all you have to do is to follow these fibers back to where they originate in the retina. So let's follow the green ones first. So this particular fibers here, these axons, they travel on the same side, and they originate in the left temporal retinal field, which detects vision in the left medial vision field. Left medial vision field. It's affected. Make sense? Now, this red fiber crosses over at the optic chiasm and originates in the right nasal retinal field, which detects vision in the right lateral visual field. So therefore, right lateral visual field. is affected. Okay? So, in this particular case, what do you think the patient is going to report to you? It's a weird.
I can see on my right side.
So let's say if they're looking straight, can they see?
Yeah.
Yeah, with both eyes open, they can see. A little better toward the left side, a little worse toward the right side. Because remember, the vision actually blends in the middle somewhere, right? Well, we're not going to talk about that too much. So now, if you ask the patient to look toward the left, they can see fine. Look toward the right, poof, all of a sudden everything just kind of goes dark. Or at least really grainy and gray and stuff like that, okay? Look up, look down, same thing, it's just the right side, it's a little bit of a haze, okay? So we call this homonymous hemianopsia. Homonymous means it's on the same side in both eyes. Hemianopsia because each of these visual fields are only half affected, okay? So you can say right or left, in this case it's more right homonymous, right? Again, hemianopsia. These are the three basic ones you guys need to know. Okay, so go home, draw this out, and cut in every possible way. Don't cut this way. I mean, you can if you want. It's just black. Black out altogether, right? This is total blind. All right, good?
Can you have only
Yeah, you can. You definitely can. But we're not gonna talk about it.
what would cause only half a cut?
So let's say if you have some kind of trauma, and that trauma for some reason, like, sorry guys, go for take. So let's say if there's a trauma right here, and you only shear this fiber, and you can have a visual defect like that. Absolutely. Or just a little bit of the optic tract, and not completely. But then how do you know? Right? Because all these fibers kind of blend in together. It's not like they're separate the way I drew it. Right? It's only here that it's clear cut where it crosses over with this one. All right, hold on, we're not done. Because I'm going to give you two other ones for you guys to figure out. Okay? So if I make a cut here, what do you think it's going to look like? So the defect now is in the left inferior tract of the optic radiation. Okay, so now what we gotta do, again with the visual fields, we're going to divide into right and left or medial lateral. But because these are the optic radiation, you have to now divide it into superior and inferior. So now there's quadrants. Okay, so it's the same thing. It's actually not as hard as you guys think it would be. Right? It just seems complicated because now it's divided into superior versus inferior. So these two are synapsing with those two. Those two are basically traveling in the same pathway as if you cut the optic tract, right? So for this inferior tract, this one follows the green line all the way up to the left temporal retinal field, which detects vision in the left medial. Left medial, okay? But because that's the inferior tract, then only the superior portion is affected. Okay? And then this particular fiber here, following synapsing with the red, follows it all the way back to the right nasal retinal field, which detects vision in the right lateral visual field. But because it's the inferior tract, right lateral is over here, it only carries information about the superior area of the visual field. So because the inferior tracts are affected, the superior quadrant on the right side is the only one that's affected. Right? Kind of a unique visual defect because the patient can actually see fine for the most part, but if you ask the patient to look up toward the right, they can't see much. Right? So again, you can say this is right versus left. So say left, right. Of course, this is going to be right, right, and this is going to be superior. This is superior homonymous. Quadrantopia. Okay, quadrant because there's 4. Right. All right. Here's the last cut. That's number 5. Something's happening at the visual cortex. What do you think?
Right inferior medial.
Okay, so let's say if all of these are affected, you would actually have total blindness.
Total blindness. Oh, I thought did you cut all of them out? Oh, I thought that you only cut like only the the
specific
ones in the
ones?
middle.
Yeah.
And there are like 4 around.
But, you guys remember the video of that individual? I don't even remember his name. Colin? No, Colin's the guy.
Oh, the one that reacts to scent movement.
That's right. Yeah, he can't see, right? But he can tell if the object is moving. If it is moving. Why? Because there's another pathway called the tactile pathway. And the tactile pathway is for visual, sort of like reflexes. Remember the pathway I drew out to you guys where you shine a light into a pupil's eyes and the pupils like constrict and stuff like that, right? It goes to the midbrain and goes to the oculomotor nerve and stuff like that. That's the tactile pathway. Okay? That is a reflexive pathway for visual information. So which means that these particular individuals, they have total blindness. Except for one portion. So we call this total blindness. With what we call macula or macular sparing. Sparing. 1 R, 2 R. Sparring.
So the cut was made in the optic house.
Yeah,
and
just
the
think
exceptions
like,
were
let's say
not
the patient
exceptions.
had a stroke in the occipital lobe, in the visual cortex. Yeah. Yeah, so all of those fibers are affected in a sense. So they'll have total blindness. This is like the Daredevil. You guys ever seen Daredevil, like the DC superhero? He's totally blind, right? But he can still see. So if you want to fight the Daredevil and beat him, just stand still. He can't see you. If you move, he's gonna kick your ass, because he can see your movement. But if you stand still, you're blind to him. Right? And that's why in a gunfight, I don't know why I'm talking about this. You guys ever watch like Wyatt Earp and like Tombstone and OK Corral? Oh man, this is such a good movie. Anyhow, back in the days in the Western era, supposedly it happened where these particular, Westerners, they were approaching a gang and they got into a shootout, okay? Now, this main character, his name is Wyatt Earp. He was super calm and cool, and when the chaos erupted, he stood still and he basically drew his weapon and started shooting people. But the bullet never hits him. Because as you are in that super, like, really high intense sympathetic kick, you don't really see much, you're just seeing the reactive stuff. So when people are moving around, you can actually shoot at those people. But when the person is standing still, that person is virtually invisible. And that's why Wyatt was standing there, still, everybody's kind of moving around, he just basically starts shooting all these people. Anyhow, that's just the tactile pathway. Alright, any questions about visual defects?
Could you explain number 5 to me? And I don't get
Mm-hmm.
how the entire visual. Yeah.
Yeah,